Okay, guys, this is very exciting because it really gene editing and genomic modifications of cells has been really making incredible progress in the last couple of years. Actually, it was an explosion of labs jumping into it, and it had to do mostly with the advance of the technology. So this is kind of what we're going to see today. It's a brief overview of how I like history always, so a little bit of the history of how we come up with this, how we end up actually doing what we're doing this, why the whole world jumped into CRISPR. Now, you can stop anyone in the street and ask CRISPR, and they will say, yeah, I know that. No, they don't know exactly what is it, but they heard CRISPR, and it's been all over the news and everywhere. And of course, for us, that means that we have now more access to technologies that allow us to do what we wanted to do for many, many years, actually. It's not new what we wanted to do. And that's kind of what I'm going to show you today. And a little bit about understanding the basics of how it works. And not everything that shines is gold. So even if it says that it's easy to do and very quickly, it's not. And we're going to also discuss that. At the end of my talk, Katie, she just defended her thesis, so she's graduating. She has a lot of experience with gene editing and CRISPR. She will come and show you in real life how to design, how she does it basically when she needs to decide from the moment she wants to target a gene, how to do it, exactly the way that she does it, okay? So to give you a more even practical approach to, if you decide to do it, how you can do it. This is the definition of gene editing. We've been seeing so many definitions, right? So this is gene editing. Genome editing or gene editing is a type of genetic engineering in which DNA is inserted, deleted, or replaced at a specific site. So I'm going to emphasize this in this definition, which is similar thing to many other things we do for genome modifications. We can talk about gene therapy, what's the difference, and that's my next slide. At specific site, that's very important, in the genome of an organism or cell. So this is one of the few definitions that are out there. It's a concept that hasn't had many years, but people have been doing this for many years for very different reasons. But it became, as I said, much significant for what we do in just recent years, mainly because of the development of the technology. So this is the definition of gene editing. And as I said, specific site is what is key that differentiates gene editing or genome editing from previous concepts like this one. And I'm going to start with gene therapy because that's how I got into this field. And that's how many people got into the field of genomic modifications. And it started many years ago in the 70s already when people found ways to introduce pieces of DNA into mammalian cells. And by doing so, they were trying to find ways to introduce those pieces of DNA into the host chromosomes. That was key to try to do efficient gene therapy. But gene therapy, in slight difference from genome editing, is the introduction of a functional gene into a target cell with the purpose, and that's why it's called gene therapy, with the purpose to restore the production of a protein that is absent or deficient, mostly due to a genetic mutation. Now here, as you say, it's just the introduction of the gene. So the specific part is missing. That's what really brings us to gene editing. But we're going to talk very quickly about gene therapy because if I ask you, how can you put DNA into a mammalian cell? And there are several ways that you can think about, and people have been trying all kinds of ways, but we're not going to stop into this. I mean, you need to know that there are different ways to introduce in DNA. But I want to emphasize viral vectors because viruses have evolved over millions of years to do just that, introduce their genome into the infecting cell. And we basically take advantage of that. So people in the field of gene therapy have been using viral vectors to introduce pieces of genome. It can be DNA, it can be RNA sometimes, by taking advantage of the fact that viruses have evolved to do that. And you remember I showed you this table the last time when we were talking about different vectors. And there are three main vectors that people use today for gene therapy or for gene transfer: adenoviruses, adeno-associated viruses, and lentiviruses among retroviruses. This is the oncoretrovirus I mentioned as well, with advantages and disadvantages and different characteristics. We're not going to look into this, but I do want to mention historically one particular case. So for many years, people have been trying to find ways to introduce these genes and been developing vectors. One of the vectors that people used for many years was oncoretroviruses that I mentioned to you, the simple gamma retroviruses. And By the time they felt that the technology was good enough to go into the clinics, we were already thinking of using other vectors. But it takes many years from the idea and from the research to get into the clinics. So this team in France, directed by Alain Fischer and Marina Cavazzana Calvo, did basically the first successful gene therapy in the field of hematopoiesis. So they treated a few kids. Actually, there were 10 originally 10 patients that were treated with gene therapy using oncoretroviruses. They extracted bone marrow from the patients, infect them with the retroviruses, and put the cells back into the patient. All the kids, except for one that has an inflammation in the liver, so it was not counted as part of the trial, all the 9 kids were cured. Very successful, and it was news all over the place because, believe me, after many years of failures, mostly because of inefficiency, they were not able to get good virus infection in the right cells to make a clinical outcome. But in this case, it worked. And, and for many different reasons, because of the type of the disease that they were using, there was a very strong selection. Even if they got very few cells corrected, those cells will expand, and that's helped see an outcome. Okay? So the kids were cured, nine patients. Big news, they became instantly famous, and a few years later, so this is 2000, 3 years later, they report this. Same people. So first 2 patients, and then 4 patients out of the 9. Mm-hmm. They develop an oncovirus integration-related leukemia. So what happened? You remember I mentioned to you insertional mutagenesis, right? So there was such a strong selection that many of these kids have integrations in a very specific locus. It's called LMO2. In 3 of them, I think the 4th one has another integration. And that LMO2 is a known gene that it can become activated in leukemia. In that case, the integration of the retrovirus activated LMO2 and induced an abnormal expansion of a T-cell population, creating a T-cell leukemia. So that happened. And I think I have a slide explaining the mechanism. So this is basically the sequence, and they found where this is the virus, and this is patient 5, patient 4, patient 10, patient 7, okay, and where in the locus they're integrated. So this is the LMO2. One of them has also next to a CCD2 gene and a BMI1 gene. But all of them somehow were related to LMO2, a very strong selection for that integration. So that's bad. And this is what I was telling you that we cannot control. And that's why we don't have the specific part in gene therapy definition. We cannot really control when we use retroviruses or lentiviruses where that piece integrates. And that can cause problems. Okay? So that's one of the reasons people have been thinking about we need something better. I wish we could go into the genome and just modify one base pair or a few base pairs. And that's it, nothing else. And that's basically what gene editing is supposed to be. In parallel of all the development of gene therapy and gene transfer and gene transduction, we were developing techniques to get better at isolating pluripotent stem cells, embryonic stem cells, isolating from inner cell mass, or induced pluripotent stem cells that we created by using exogenous factors. Now, why is that relevant to gene editing? Well, stem cells self-renew, and they grow forever, literally, in culture. That means that now I have the opportunity to work with a cell that gives me weeks, if I need weeks, to do a modification. Now, in contrast to gene therapy, in the case of gene editing, there is a process where I need to select, because the efficiency is still not great, it's very low, and I need to select for those where it happens. And we're going to see what I mean by happening. But the stem cells provide me with that ideal window of opportunity, because the cells grow forever as stem cells in culture. They're not changing, they're not differentiating, if I know what I'm doing, and you're learning how to do that without differentiating this. Hopefully by now you're starting to understand and how that works. So that happened in parallel to the whole development of introduction of genetic material. So for the first time now, I have in my mind a very straightforward, amazing opportunity. So let's say I start with an iPS cell from a patient that has a mutation in a gene called HFE, which is a gene that is mutated in a disease called hemochromatosis. And this is a disease I've been studying for some time, It's a project that is still sitting there, but we started doing it, and we made some iPS cells actually from patients here, collected blood from patients with hemochromatosis here at Boston Medical Center. And that's a particular mutation that that gene has. So now I can take those iPS cells from individuals that has this mutation and correct, hopefully, this mutation, only the mutation, with some type of gene editing technology. And if that's the case, now I end up with an iPS cell that is identically to that one, except for the mutation. And if I can now, one day, make this three-dimensional liver, which we cannot do yet, then I can end up with a liver, an entire organ of the same individual that has a disease, but the liver is corrected. So that liver has no disease anymore. Very straightforward, very simple concept. This is what we want to do. And I have no doubt that we will be doing this. It will take us a few more years, I guess, to get there, but we're working on it. What you're going to learn today is the different ways and technology that we can do this part, this gene editing. And that's what we got into engineered nucleases. So we needed more specific approaches that gene therapy couldn't give us. And people have started looking into taking advantage of enzymes like meganucleases. That was the first approach, and you can barely see that in the books anymore or even reviews. They don't talk about meganucleases, but that really was one of the first attempts of getting access to specific cutting. And this is just a summary of the three most common ones. This is historically one of the first to really, really get into an efficient approach of using engineered nucleases to modify DNA, zinc finger nucleases, and then TAL effector nucleases or TALENs, and finally CRISPR-Cas9. So this is the three we're going to talk today very briefly about, mostly these two. But how that works? All of them basically do kind of the same thing, which is introduce a break in the DNA. So all we're trying to do is to try to be as specific as possible in using an enzyme or some type of machinery to create a double-stranded DNA break, or if it's single-stranded like Nick, in both places. So at the end of the day, we're trying to stimulate the cell to recognize that we are creating a DNA stranded break. And if that happens, the cell has its own way of fixing that. So there are basically mainly two ways that they will try to fix it. Either using non-homologous end joining, which is an error-prone mechanism. And we take advantage of that error-prone mechanism, because when the cell is trying to correct, it often results in the introduction of non-specific insertions or deletions. And those resulting mutations can then induce a knockout. And sometimes if that's what you're looking for, then all you need to do is this. But if you want to be more specific, for instance, and correct a mutation or introduce a piece of DNA to have like a reporter, so you create a reporter line by putting an iris GFP in a very specific site, for instance, under a specific promoter. You remember I talked yesterday about SOX2, 2 days ago. SOX2 GFP, that's kind of the way we want to do it, right? When you need, then you need homology-directed repair, or HDR. In that case, you are delivering an extra piece of sequence of DNA that contains what you want to introduce for the correction or for the insertion. And in a very low efficiency, but still happening, the cell will get confused during the repair, and will use the template that we are providing to do the repair. And then you end up, if you have a way to select, maybe it will increase your chances of finding that, a cell that now has introduced that. So that's basically what this shows. These are basically all the different outcomes, okay? Chromosomal deletion, or gene disruption if you do non-homologous end joining, gene correction, or targeted integration, which is what I just described. And this is just one example. If you sequence, when you do what the different options that you can get. So here are the different deletions that happen in one real life example of using gene editing. Okay, so we're going to jump directly into the two best examples. And I'm still going to talk about TALENs because TALENs are still used a lot. And I'm still a big defender of TALENs, because even though CRISPR got all the news and all the noise, there is no reason to forget about TALENs, which was really the first time they gave us, people in the lab, in the normal labs, access to do it ourselves. gene editing. Zinc finger nucleases were very difficult to construct. There were a lot of troubleshooting in trying to find the right construct. And so very few people can do it, and mostly were done by companies. The first time that the company did it, which was Sangamo, and Sangamo became a big player. It's a company in California, a big player in gene editing, because they were one of the few, the first that started this business of gene editing. But when they started first, and they licensed the technology of zinc finger to Sigma, and you can order for only $25,000 a pair of zinc finger nucleases. And it's interesting because the moment they started out selling that, I think a couple of months later, the papers of TALENs came out. And immediately the price dropped to $5,000 because no one wanted to buy any more zinc finger, because now you could do this. So transcription activator-like effectors, or TAL, occur naturally in plant pathogenic bacteria. That's how we find, and many of these things we find actually in very, very primitive organisms that develop their own immune system, and this is one of the type of how they defend themselves from exogenous genetic material. So the TALs contain a modular DNA binding domain comprising tandem polymorphic 34 amino acid repeats. So each one of these has 34 amino acid repeats. And within these repeats, amino acid 12 and 13, which are these two letters here, can recognize a specific base. It's kind of incredible if you think about biologically how a protein that can be so big still can recognize one base in the DNA. So we learn to know which of these amino acids can recognize basically each one of the base. So Ni combination recognizes adenine, HDC, NGT, and so on. And if you now know this, you can basically put together specific modules to recognize a specific sequence. Again, very straightforward, especially because the people that designed this, they did a great job at making our life easier. All you need to do, it's a multiple, one-step multiple ligation to put all of these pieces together. And that's how it worked. It's kind of incredible. But when you do that, and you fuse then the TAL effectors to a DNA nucleus, in this case, it's called FOC1, and that's the same one that people use for zinc finger nucleases. Then it became TALENs. Okay, this is the TAL together with the enzyme, the endonuclease. Okay, and that's basically, it's very relatively straightforward, and most people can do it in their own labs. This is exactly how they look, basically. And because of the way that it's designed, the nuclease domain needs to be dimerized to work. And the reason they did it, which is clever also, comes from the same concept in zinc finger nucleases. So that was not new. So we learned a lot with zinc finger nucleases, thanks mostly to this company and a few other groups. But the same concept was used for TALENs, and they need to dimerize to really work this FOC1 nuclease. So it doesn't cut unless they dimerize. But by doing this dimerization, now you increase by twofold your specificity of the sequence, because now we have two of these TALENs binding to the DNA for then inducing the DNA double-stranded break here by the FOC1. And that's how it is. So you design normally two TALENs, one for each side of the sequence that you want to cut, and you put it together in this multiple ligation reaction. Now you can buy the whole plate with the different bacteria that you amplify, and then just do it. It's really incredible. But not everything that shines is gold. It worked, and it clearly cut, but you need to test a few of these things. I think that's a golden gate kit, it's called, that you can get it from Adgene. And it's really, as I said, it comes in a 96 well plate that you amplify the bacteria and you get all the different pieces. So, how do you know if it works? There is one technique that a lot of people like to use. It's called surveyor nuclease assay, which is, again, not great. And I didn't mention yet what's the problem with TALENs, but here is one example. The way the surveyor basically assay works is it's a mismatch sensitive assay. So what it does is a PCR that can detect mismatches. And if you do cut, those mismatches will appear as different bands. And you can kind of calculate the efficiency of your cut by quantification of the other bands. So you know what the wild type band should be. In this case, it's the upper band. And any lower band will be a proof that your combination of talents are working or cutting. And you can kind of calculate efficiency, percentage of efficiency of cutting by measuring the density of the band. It's a very crude assay. It works. It's always an underestimate because it only detects mismatch. That means that when there is a mutation, you denature the DNA, renature, and when there is a mismatch, that's where an enzyme will cut and form these bands. But if the mismatch happens to put together two of the same, you won't detect, even if it's cut, you won't see this in this assay. So it's always an underestimate, which is fine, I guess, because all you need is to see here cuts. But it's very specific for cell type. Most people use Surveyor in a 293 cell assay when you're going to use your talents mostly in IPS or ES. Then it's not really great, but it's a first step if you want to have an idea of how your talents are cutting. And for talents was, I think, more relevant and more important than for CRISPRs, and I will explain why. But for CRISPRs, people are still using also the Surveyor to give an idea of how good the CRISPRs cut. Now, this is one gel, one example. Here we were trying to mutate the APC gene. This is a gene that is very important in colorectal cancer. And we have a project on colorectal cancer and iPSL modeling of that. In any case, we were trying to use talents to induce a mutation, create a mutation in APC. And we combined 3 different type of pairs. And by doing, and this is one just control, HFE, that I mentioned before, that another gene. And you can see here, out of these 6 different combinations, one really gave me a very strong band. And this is typical of talents. And that was kind of one of the issues with talents. There was this need of creating several pairs and testing because there was no real way of knowing which one will cut well. And there were a lot of arguments and discussions why mechanistically that was the case, even though everything is designed perfectly well. It's recognizing a piece of DNA the same way. Some of them cut better than others. And that was kind of a problem because it means you need a little bit more work, but really not that much. But you know, at the time that we were trying to learn this and optimize it and understand it, then CRISPR came out. So this is CRISPR, okay? Cluster, regularly, interspace, short, palindromic repeats, or CRISPR for short. And I was reading yesterday an article about this, and it's interesting because everyone knows the technique by CRISPR. And CRISPR actually doesn't do anything. CRISPR itself doesn't cut, doesn't ligate, it just repeats. That's CRISPR. But everyone knows CRISPRs, like that's the technique. And in reality, you always need to say CRISPR-Cas, because Cas is what really does the job. Cas means CRISPR's associated proteins, okay? And this is really the technology that changed gene editing in many regards. I would like to believe that yet, for many purposes, and we're still using talents in the lab, and people have created their own talents that worked. Those talents are still perfectly okay. You don't need to go and do CRISPRs now if your talents are working. But it really made the technology maybe a log faster, easier, more efficient. And by that, it changed how people in the labs approach it for many different reasons, for many different applications, for many different purposes. Even applications where talents were not practical, were not efficient enough, CRISPR, boom, worked. But as I said, and we're going to discuss this, you know, it's not that straightforward. You still need to do quite a lot of work to get it done. But how that works, and there is one major difference compare between zinc fingers and talents, because in zinc fingers and talents, both of them has a protein component that is recognizing the DNA sequence bound to or chimerized to this FOC1 nuclease. CRISPR-Cas9 doesn't have a protein and doesn't have the FOC1 nuclease. The CRISPR-Cas9 system works slightly differently. They have a guide RNA piece. This is an RNA template, which is homologous to the piece of DNA that you want to target. And when that RNA basically recognizes its template, then these CRISPR-associated proteins, Cas, come and do the job of cutting and ligating. And so the idea is still the same. You are inducing a double-stranded break, and you're trying to induce the cell to do either non-homologous enjoining or homologous repair base. Okay? Now I want to ask you, who here is familiar with mouse knock-in technology? which, by the way, got the Nobel Prize, okay? Mario Capecchi. You know that, you know that, right? That, I mean, people have been using mouse knock-in technology for many, many years. And the basis is kind of similarly. You induce a double-stranded break. But in this case, and people have done this in mouse embryonic stem cells, so mouse ES cells, you transfect a piece of DNA, and you expect that to happen. Now, the reason we needed to go into gene editing is because in human cells, the efficiency of that homologous base repair is much, much lower than in murine cells. In murine cells, we don't need gene editing for that to happen. And it's efficient enough that after selection, we can find clones of these murine ES cells that have the right target. In human cells, it never worked as well. I mean, there is, I think, one report that really managed to do it. It was, again, very, very low efficiency and not practical. So people were looking into this technology as a way to improve the efficiency of doing knock-ins and knockouts in human cells, because it couldn't do it without the help of creating the double-stranded break. So all it is basically in improving the efficiency of that double-stranded break for the cell then to do repair. Okay? So as I said, there is the way that we now do it. You construct a vector containing that guide RNA and a promoter to induce expression of that guide RNA. And that will also bring now the optimized Cas1. Cas9 is one type of Cas, so they tested many different ones. It was originally found, so if you look in historically, it was the dairy industry. So people working with milk and yogurt, they had trouble because they started finding that their milk was getting acidified too much. And they couldn't understand exactly. Anyway, when they started looking, they found these repeats, and they found that it was apparently coming from the presence, I think I have this, no, of bacteria. So this is basically an adaptive immune system of bacteria and some archaea. And these bacteria will try to degrade invading viral and plasmid DNA. The bacteria uses this CRISPR to prevent re-entrance of a virus or plasmid DNA. Okay, so the repeats, I think we have one more person coming. So good luck finding a chair, yeah. Marianne, maybe you can sit there. So when they were trying to understand what was happening, they found these repeats which were not part of the bacterial DNA. But the bacteria kept these repeats as reminders, memory of this invading DNA. So it was very quickly easy using this guide RNA, recognizing if it was trying to invade again. That's basically what it was. And that's how it started years ago in the 80s, describing these CRISPRs. Okay? There is a lot of discussions, and we're going to have a brief discussion about it. Lots of discussions about who really did it first. Now, if you ask me, I would say, why it's so important? But apparently it is important for a lot of people. I would like to believe that that's not the reason, but there is a big monetary interest behind the discovery and the patent and the license. And I don't know if you've been following those news. I will tell you a little bit of what will happen. But I really like this. You know, this is from a review by Jennifer Doudna and Emmanuel Charpentier, and we're going to already mention them. Because at the end of the day, I think most of the scientific field accepts that Jennifer and Emmanuel were the first to really bring the technology to be useful. They really demonstrated for the first time that you can manipulate that to modified mammalian genomic DNA. And that was in 2012. That's their paper. And I'm going to show that. But as you can see, historically, you can really go back to 1987 for the first time that CRISPRs were described. And here they use this nice comparison timeline between gene editing and CRISPR biology to see how they converge here around 2011-12 to really start making, and these are papers. Each ball is a paper, basically. And it's an explosion. Actually, yesterday, if you PubMed now CRISPR, you put CRISPR-Cas word, there is 1,900 papers, which is a lot, believe me, for 3 years, 4 years. Anyway, so this is really the key one. That was a science paper, August 2012. programmable dual RNA guided DNA endonuclease, CRISPR. Okay, and they say basically they've been studying bacteria and RNA modification for many years. These two groups. So that was kind of an interesting finding for them. They, as they say it themselves, they never imagined that it will cause such an amazing impact in the field of gene editing and stem cells. A scientist from MIT was in a meeting listening to this. And he said, wow, this is really incredible if we could use this to modify human cells. So we cannot forget this paper. This is interesting. Most of the reviews or the discussion always forget this. Now, Genec I think it's a postdoc in Jennifer Doudna's lab, and they rushed to publish this paper in eLife, and that was January 2013. And I think the title is clear, right? It's genome editing in human cells. But then February, this is January, February 2013, 2 science papers back to back, one from Feng Zhang, amazing scientist. Amazing work comes always out of this lab. I mean, if you have time, you can PubMed Feng. You will learn a lot about if you're interested in gene editing, because it's really incredible work that they do. And he really, I mean, I think everyone recognizes he really pushed the field forward by the incredible work he did on developing and improving the technology. But this is the first paper really in Science where he published the description of using CRISPR-Cas9 even in a multiplex genome engineering. That means multiple genes at the same time in that Science paper in human cells. Back to back with this guy, you may know, George Church, amazing scientist also from Harvard, who also used RNA guided, which is a CRISPR, to modify human cells. So this basically is the basis for all the discussions. Who did it first? It's kind of very clear. They claim that this, which is true, didn't do any human modifications, so they couldn't do human modifications. They claim, well, this is kind of not true. We already developed the technology and modify mammalian cells. It's kind of one more step. So there was a big discussion for many years. I don't need to tell you, but there were multiple companies opened after these discoveries. And a few months ago, the decision by the court was that Jennifer Doudna and Emmanuel have the license and the patent rights for CRISPR technology, and Feng Zhang keep the rights for licensing the technology to use CRISPR for human cells. So of course they are appealing this. They say that's not fair. It's like I like that comparison. They say it's like we have the license for all tennis balls and they got it for yellow balls, which is a good comparison. It is true. So basically they were very happy. They were not that happy. And we are all very happy because that technology is what we need, okay? And patients should be very, very happy because we will be bringing this technology to patients one day. Anyway, since the original discovery, there have been several modifications trying to improve the way CRISPRs work. If I think, stop 10 scientists here in the street and ask them, are you happy with CRISPRs? Marianne, what are they going to say? What do you think? I think that half of them will tell you, I am very, very happy. Trying to make the case that maybe you don't know what you're doing if you say no. The other 5 will probably say, oh, I'm also, I guess, very happy. But the reality is, I can tell you with my own hands, right? We did that in our lab. We're still trying to do it. It's not that straightforward. So we also been working with tailends before. So we have even the comparison. I have a slide to show you that. But overall number seems to suggest that it's better. And that's real. That is real. There's still a process in doing it, and I think that you're going to hear that more from Katie. And you can ask Katie what she thinks about it. And when it works, it's amazing. When it works, it works. But I will argue the same thing for tailends. When tailends work, it works. And it's very fast and it takes you a month and a half to have a clone that is basically targeted. But anyway, because of that need to make it better, more efficient, there has been coming up with these improved versions of it. This is one example. I'm going to give you name 2 of these examples of improvements of the technique, it's very hard for me personally to really get a grasp of if there is a need or not to move into these new technologies. So there is a third group I didn't mention, and I want to mention that works also in MGH, in Harvard affiliated, Keith Jung, who also he spent so many years improving tail ends and zinc finger nucleases first. So for him, I think CRISPR was kind of an issue, but he managed to get also into the CRISPR field, and I think he's also very happy now, Keith. And I have conversation with Keith trying to really understand, because every time, this is real, in the year that this technology came out of CRISPRs, we were starting to set up the reagents, and a new paper will come out suggesting that maybe we shouldn't use this CRISPR, because now there is this new CRISPR that does nicking, and they claim is better. So in this paper, I'm going to show you basically what they did. They modified this slight mutation in the Cas, so it doesn't do a double-stranded break, it does a nick. Now, if you do now a nick, you can design your CRISPR, the nick mutase version of it, to have now two nick very close to each other. But what it does basically, it brings now two Cas9 to two different sequences. And by doing this, the claim, and it makes sense, biologically it makes sense, it's kind of the dimeric way of the FOC1 for the tail ends. It's improved the efficiency. So you are fighting against, you know what is the problem with all these endonucleases, and every reviewer will use that when they want to criticize your grant, off-target effect. So this whole field of endonucleases have one small problem, which is what happens if your endonuclease is not cutting where it should cut, and that's called off-target effect. You can measure that, and again, there is a whole world of people working into demonstrating how much or how little off-target effect their own CRISPR system does. And I think it's fair to say today that overall, from the beginning, the technology was already so good that off-target effect was minimal. I think that for most purposes of what we're doing, for most purposes, even if there is little off-target, it's not a big deal. I know that a lot of people would disagree with me. But in reality, it's if we're trying to do drug screening, and I need a very good targeted hepatocyte line, and all I'm going to do is drug screening. So I will argue that even if there is a few mutations there here and there, if that line is doing what I need to do, which is to react to drugs in a specific way, then it's fine. But if we do need, we can do now whole genome sequencing. So for clinical applications where we will need to see really if we are creating something that we don't want, we can just do whole genome sequencing and prove that. People have been using some tricks instead of doing whole genome sequencing, which is much more expensive, just sequencing the few pieces that seems to have closer homology to the potential target sites of the design of the CRISPR, and you amplify and sequence that and see, are you creating off-target effects? And I think that it's fair to say that most papers that look into it show that the efficiency of the occurrence of off-target is very, very low, actually. And I also have one more thing to say about it. And I like to think that we, living machines, all the time are mutating. We are mutating all the time. But we have very potent machineries in our cells to defend ourselves from those mutations. And those cells that we are working also have that. So you remember I mentioned that in iPS cells can happen, or in ES cells, that at the beginning, the few passages cells tend to be unstable. And they will select, that was a great paper that showed that the cells will select against mutations that are bad for the cell, because those cells will end up either dying or going into apoptosis, right? So the same thing for our mutation, and I would like to believe also when we induce bad things when we do gene editing. But this is one example of how they in this paper of the double nick, how they basically, and this was a Cell paper. A lot of these papers came out in Cell and Nature and Science. So this is how they checked. So they use the Cas9, that's a normal Cas9. This is a mutated Cas9 with nickase, and they show basically that when you put your specific guide RNA, and your efficiency of cleavage is similar, kind of similar, 24 against 25 or 32. So the nickase work as well as the other one. But when you check for off-target effect, and here it is, very, very little mutations. So this is one example where they choose 5 sequences where they were potentially off target, and they sequence that and they can also estimate the efficiency, and it basically goes from 10 to very, very little of any. I think the Cas9 nickase is this one, right? So it's almost undetectable, at least in the gel, against almost 10%, they say, when you use the normal Cas9. So according to this paper, of course, it's very important to use the. I need to check. I haven't talked to Feng in a long time. So I don't know what his lab is using right now. Probably is using the double nick system. I did talk to Keith Young at some point, because at the same time that this cell paper came out, he came out with a Nature Biotech paper, I believe, showing that was not necessary to use the double nickase, that he has another way to improve the efficiency without off target. But what I do want to mention was this. So almost the entire cell paper is done on gene editing on 293 cells. Okay? So most of the figures, most of the efficiencies are all done on 293. But they have one figure. This is a human ESL line. So we were talking about 25, 24%, 32%. How about the efficiency in human ESLs in this cell paper? Do you see these numbers here? It's slightly lower, right? So let's say it says here 0.4. 0.4% means how many colonies you need to check to find the right one? Out of 1,000? Yeah, 0.4, right? So you still need to pick a lot of colonies according to this. But fair to say, we have become better than this, and they have become, and everyone became better. That was one of the first few papers to show this, and the efficiency was still low. We still need to do a lot of things to improve efficiency and to select the clones better. This is still an issue, and we're still dealing with that, how to get it better. And this is another example. So that's another cell paper, okay? And this is all really matters of months. In a few months, we have the double nick, and we are all happy, and we're going to maybe use now the double nicking. And then it comes with CPF1, which is a different type of Cas, same family, type 2. And I think I have only one figure, but just to exemplify. It basically has a few differences compared to the Cas9. It works in a similar way, but it uses a single guide RNA. The other one was also combined. It was two, but it was combined in one. So for me, it's not that much different. But it has a slightly different sequence recognition of PAM. I'm not going to talk about the PAM because I think Katie is going to talk about the PAM. But it's an important 3-nucleotide sequence that when you design your CRISPR, you need to take into consideration. And they claim in this paper, this is really great. You should all now move to CPF1. We just got the plasmids for the other one. And then no, we need to contact again AdGene, I guess. But it is all available in AdGene. CPF1, I think 3 months later, Charpentier's lab published a paper showing that this is not really exactly how it works, CPF1. And we are trying to just edit a gene. So this is a short class today. This is basically what I want you really to take out of this. And now I have a few fun examples, okay, of what's going on. But before, what about comparing? So people wanted really to know, is that better or not? And there are a couple of groups that did it really nicely. One of these is a good friend of ours, Chad Cowan, working with Kiran Musunuru, and where they really did a very large comparison. And there is a history, like almost always, behind this paper. Chad was basically targeting, like, I don't know, 50 different loci using TALENs. And they had the paper ready, and they submitted for publication when everyone started talking about CRISPRs. So he put his student very quickly to say, okay, I want you to now replicate everything you did with TALENs with CRISPRs. He did publish the paper with TALENs, but then he came out with this paper because he had everything done, side by side comparison, okay, of TALENs versus. So show a few examples, okay? Efficiency with TALENs, mutants per clone screen, 10%, 3.5, 12, 33, 0, 30, 22, 10, 1.6. Now if you go into CRISPRs, they go into 60, 66, 78, 66, 51, 57, 68, 75, 10 in a clone there was 1. And this is heterozygous mutations, 70%. You can get even up to 12, 7, 11, 25% of homozygous mutations using CRISPR. That was their data. And now if you look at this, it's incredible. It is incredible. That, I mean, 70% means all you need is like 2, 3 clones. One of them will be right. This is not our experience. And I have long conversations with Chad about this, Chad Cohen. And reality is, of course, when you know what you're doing and you become an expert in it, apparently that makes things, like always, better and easier. If you're learning how to do this, or if you need, you're still going to need to screen many clones. And I think there are different ways of improving this. People have been using selection with fluorescence, like GFP, and you sort for the cells after you transfect. How you transfect your plasmid is a whole world. People use electroporation, people use nucleofection. They claim, the ones that use nucleofection claims that if you don't use nucleofection, it doesn't work. Nucleofection kills a lot of cells, so you need to start a very large number of cells. So all these nuances in the technique. But at the end of the day, I mean, the data is the data, and it's real. And we can do this. And obviously, according to these numbers, CRISPRs are better than TALENs. Again, and we have a very, very small example because we were just doing for our, as I say, for our HFE gene, and we were just finishing doing the comparison of TALENs for HFE and APC. And at that time, I told Andrea, at that time it was in my lab, why don't you just try a couple of CRISPRs? And she just designed 2 CRISPRs, and both of them cut. In contrast to the TALENs that we need to try like 6 to find one that maybe cut. So even in that small example, it was kind of very obvious that CRISPR seems to be better. And I would argue, most of the time you don't even need to do the surveyor, because if you do design 2 guides RNA, one of them will work. Now there has a lot of discussions about, if you ask now Chad today, I want to knock out this thing, how should I do it? He will tell you to design 5 guides, 5, and put all together. You will get a knockout. But then I talked to Andrea, who is now working in a company doing gene editing. All she does is that. And she said that's not convenient, because it's very difficult then to, when you do normal sequencing, to really analyze the histogram. And you're always going to end up doing topocloning to analyze biallelic sequencing, each allele independently. If you just do one guide, only one, almost always cut. You just pick a few clones, 24, and sequence them. And you will find a few clones that, because it has only one guide, it will have 1 base pair deletion. Always find it. That's what you need if you want to make a knockout. Knock-ins are still a little more complicated. And now I know as a fact that biologically, homologous base repair is just much less efficient than non-homologous end joining. It kind of makes sense, but nobody talks about it, in meetings. Nobody talks about the efficiency of homologous repair base compared to non-homologous end joining. But it's a fact. Okay? So, again, CRISPRs are faster to do, high efficiency, and maybe has lower specificity than TALENs. There's a lot of arguments about that. The double nick claims that they have high efficiency as TALENs. At the end of the day, the data suggests that both of them are fine. Very little off target. Okay, doesn't this look like the Oscars? I mean, come on. But it's not. So this is Charpentier and Doudna. They received the Life Science Award. Do you know what this award is? A few young, very rich people, including Facebook guy, they get together and they decide, they want to decide who are the best scientists. So instead of creating a Nobel Prize, they say, Nobel Prize are decided by Sweden. We need something here. So they put a lot of money together, and now every year they give away 6, I think, awards per year, $3 million, called the Life Science Award. And Emmanuel Charpentier and Jennifer Doudna got that award. That was a year and a half ago. And everyone was very happy, and they were very happy, and they dressed very nicely to get this. So what are we using? And so everyone basically knows that they're going to get the Nobel Prize very soon, actually, because of the impact that CRISPR has. So what is gene editing now used for? Okay. So a group in South Korea, together with Chinese scientists, used this to create what is called double meat pigs. Now this is a very important thing, because they want to have now pigs with double the amount of beef, double the amount of meat. And they use gene editing to knock out very easily. And this is something they say that you could have done it, and people have been doing it in cows by just breeding. It takes you like 30 years to get here in one attempt. They mutated myostatin, which inhibits growth and muscle cells growth, and they got these animals. Now, it's important to remember, this was never published. They, I think, obtained around 20-something piglets. Out of those, only a bunch were born alive. And out of those, I think 3 got to be 8 months old. And they were not completely healthy. And this is a picture. And they claim, based on the picture, that they have what they're looking for, the trend to be double muscle. So that's one example. At the same time, there was a paper in Cell Research. This is a lab in China where it's multiplex in monkeys. So they tried to knock out 3 genes, but they got a pair of twins that were born with 2 mutations, 2 loci. The third didn't work, but 2 of them worked, PPAR gamma and RAG. And you're going to ask me, why they were doing this? Well, not exactly known. There was not really a biological purpose. So those monkeys are immunodeficient because they have a RAG mutation, but also a PPAR gamma, which is important in different pathways, including inflammatory pathways. But they just wanted to show that now in high mammals, these are monkeys, by injecting the CRISPR directly into the embryos, they can induce mutations. That was done. And this is where people now are trying to claim that you can do it. So not that long ago, this is George Church. It's very easy to see him when you see him in the street. And it's a very tall guy, amazing scientist. He did a lot of things. I mean, this is only a game for him, the CRISPRs. He's the one that changed the way we sequence DNA, make it like much cheaper, and that's what we can do now, sequencing very, very cheap, one of his discoveries. But what he claims now is that they're trying to knock out simultaneously all genes that are in pigs that can be recognized by the human immune system, like 20-something genes or 60 genes. And they claim they can do it now, and they already did it. It's still not published. But that's another example, because they say that, and they open a company to do this, where they will be creating organs in pigs that are basically immunotolerant for us. So they're going to use these organs in pigs for transplantation in humans. Another great example. I mean, the idea is great, but it's still not published. Now, and this is from 2 years ago, what actually species and for what purposes people have been already used Cas9. This is specifically Cas9. It's even more if you count the other systems like the CPF1. But many cell lines, many different model organisms. It's going to be used for crops, plants, very important actually. Now, how many animal products right now we have in the US that are FDA genetically modified animals that we can eat, approved by FDA? How many? 0. So you probably read about, there is a salmon. I think it's salmon that has a modification to improve the colors. It's still not approved. I think it's been like 10 years they've been fighting, and they still cannot get through the bureaucratic part. So that's already interesting. But we've been using, people are using CRISPR for a lot of things. Now I'm going to give you one last example, where we are doing it, okay? We like disease correction. I think I mentioned here, and you're going to see this a lot in the CREM. Most of the labs here are using iPSCs to model diseases. This is a paper that just came out from Katie, who is going to come into a few minutes to give you the second part. An amazing paper, Finn Hawkins, and this is Daryl, in cell stem cell, describing basically the derivation. And Daryl mentioned, I think you asked about how far you can go in differentiation. This was that paper that he was mentioning about one step farther. They can now differentiate into obtaining like distal or airway organoids. And there is one particular disease that the group is very, very interested in, is cystic fibrosis. Cystic fibrosis affects many different systems and many different organs, but lungs are particularly problematic for these patients. And the mutation that these patients have is in a gene called CFTR, which is a channel, ion channel for chloride in the cells. And when you don't have that channel functioning, you start accumulating, for instance, secretions. And in the lung, it gets very, very bad, and also in the airway. Okay, so they wanted to see if they can model this disease. So they made iPS cells from patients with CFTR mutation. There is one mutation that is the most common one called delta F508. And they took those iPS cells and they corrected the mutation with CRISPRs. Can you see that correction? Well, they developed a system that was published already before where you can make these organoids, okay, and these are airway organoids, and you can put them in culture with a drug called forced calling. Now, forced calling induces swelling because it activates that receptor and induces swelling of these organoids. In this particular case, this is the movie, 24 hours of a movie of the airway organoids from a non-corrected. Okay? So let's see what happens after 24 hours. Well, not much. And you can measure, actually. They have all the quantification of expansion of these organoids. So 24 hours, not much. Now they take the same exact line, the same exact iPS line, same human being, but they corrected the mutation with CRISPR, with gene editing. And they expose now the organoids to forced calling. Let's see what happens. Boom. There you go. Picture, 1,000 words, movie, 1,000 pictures. Nice, huh? So that's basically biology working. And if you can use this, you can now test drugs that will do the same thing as forced calling that cannot be used in clinics. Okay? So this is it, guys. This is the real life thing that we're doing now these days with CRISPR. So it works. It takes time. It takes a couple of months maybe, and in some cases a little bit longer, but it works, and you can do it. And now, you remember that figure? iPS cell going, mutation, correcting, now maybe a liver. Well, these are airway organoids that you can use now to test drugs and find new treatments for these patients. One day, maybe, we will also build lungs. I think this is it, yes. So, breathe. We have, wow, 10. A few minutes. I think Katie is on her way here, so I'm going to go grab her. Questions? Either you understood or not. I mean, one of the. So the students, you don't need to stay, but you are welcome to stay if you want to hear this. It's going to be very short, I think. It's a practical example of how to do the design. Okay, so let me grab Katie. Okay, so no questions? Yes?
Have you ever tried to use the CRISPR-Cas9 combination with the Cre and Flox to editing the animal cells, or never?
How you will do that?
No, I'm just guessing. So because as we know that.
Well, yeah, the issue with Cre, but the Cre-Lox is kind of the same thing. The Cre-Lox you need to, if you want to do it in the genome, Cre-Lox, which people do, you need to introduce it first.
Yeah,
anyway.
I mean, during the process of introducing, can we just combine with the CRISPR-Cas to introduce the Cre system into
we kind of do it when we do knock-ins. Yeah. Because most times there are pieces there of where you're putting for selection that you don't want to keep forever. Those pieces are already LOXP flanked. So for that purpose, you are kind of already doing it. But again, the whole issue here is we need to induce the double-stranded break to promote more efficient homologous repair for knock-ins, mostly in reporters. That's key. And that doesn't happen unless you're doing gene editing, okay, in human cells.
How do you introduce the CRISPR-Cas into the cell?
Literally, how you introduce that in the cell? I think Katie is going to say that. Okay. We use here nucleofection, and we also used to use electroporation for some time. And again, there is this good and bad for both of them. I think most people now using nucleofection here. The nucleofector costs $15,000, so that's a limitation if you don't have a nucleofector. But most groups use nucleofection today. You still need selection. It's much better. Without selection, then you're going to be screening a lot of colonies that are false positive. Yeah. You can understand that part. So you put something that has puromycin selection, and you do puro selection on the cells, and you have 24 clones arising, growing nicely. You pick them, you check, they are all false positive. Wait, they are puro selected. So they are resistant to puromycin. What happened? Well, DNA gets in. Apparently, somehow. in some efficiency that is enough that you capture, because you are selecting out of 1 million cells a few colonies, right? So that's why selection using either the pure itself or GFP helps. But you still need to screen, because many of them are false positives. So people use diphtheria toxin, which is an old trick. You have a piece of diphtheria toxin that it only gets it gets inactivated when the construct get into the right side. When it's in the wrong loci, it's killing the cells. So it kind of helps. But many people claim that when you have that, your efficiency drops so dramatically that you can barely find any clones. So then a lot of people don't like to have diphtheria toxin. So these are all practical things that you need to decide when you're designing your strategy. Let me bring Katie.
In the mouse.
Marianne, you want to call Katie?
In the mouse, then, is worth using the CRISPR or?
Oh, in the mouse
they
just did a
regular
transfection, regular transfection, and somehow in mice during cell division, the finding the template just by homology. The homology repair method is much higher efficiency than in human cells, and it happens.
Yeah, but then it makes sense to use CRISPR in mouse, or
no, in mouse
doesn't increase any the efficiency.
Maybe, I guess it should.
Still,
but people
you have
don't
to
use
screen 100 or 500 clones, same way as the traditional method.
Yeah, but somehow in the mouse field, I know that was never a real limitation. You just spend some time.
Yeah.
The problem in the human system is that even though you will screen 500, you will not find one. So I think that that's the main reason why people are not using gene editing for mouse, because you will eventually find it for most cases. And there are differences between different loci. That's another issue. There are clearly there are differences. And that might have to do with the accessibility, chromatin accessibility. But there are certain loci that are very difficult to target, and some that are very easy to target. And that's also an issue.
For your CRISPR knock-ins, are you using vector repair constructs or single?
That's a great question. Are you using oligonucleotides or vectors? So when you need an insertion of a big piece, of course you need a vector. Yeah. For single base pair corrections, lots of groups are using just single-stranded oligo DNA. The oligos work very, very well, are very efficient. Yeah. You still need to do the screening, but that's what most people are using.
Okay. And you're using
and
this?
We also have been using oligos, yes, for certain projects. And in some cases work, some cases not.
Yeah.
When you're introducing a new piece of DNA, how big of a problem is silencing at a particular locus? Is it dependent on the gene that you're targeting, or what you're introducing, or both?
I think it's a combination of both. We know a lot about silencing, again, from the gene therapy field. So when people were using viral vectors, that very quickly it became very obvious that where they were introducing, even though they can detect the integration by southern blot, so the thing was there, was not expressing. So when they start looking into this, they found that the mammalian cells tend to silence exogenous sequences by recognizing particular sequences that they know as exogenous. In this case, we're recognizing different sequences in the LTRs or in the enhancers of these viruses, and they tend to induce methylation. And by doing and that was one of the original, actually, one of the first paper of Rudolf Jenisch, when he was a postdoc, was using integration of retroviruses in the DNA, and he described that, and that basically people claim is the beginning of epigenetics, because he saw that methylation of these viral sequences induced silencing. So that's one example of what you are putting obviously has to do with silencing, depending on what sequences, what elements are active. That's one thing. There are certain loci that tend to be prone to more silences or less. The famous AAV-S1 locus, which is a locus where normal AAV, adeno-associated viruses, used to integrate. Now we engineer AAV so they don't integrate mostly. But that locus, basically, it's a very good locus because it's known to be very, very low prone for silencing. So people use it a lot to introduce things because in general, it never gets silenced. So it's a combination of both. In our experience, lentiviruses in mouse cells tends to get silenced very little, very little, but lentices in human cells tend to get silenced more, in my experience.